Welcome at the EUREOS Scientific Symposium. And instead of a symposium at a hotel in central Amsterdam during UGW, we have resorted to a virtual meeting. And I guess we have to look at things from the bright side. We have over 100 registrations for the symposium, several of them from outside Europe. And I think that also brings opportunities here of working closely together with colleagues from outside Europe. And also it seems it is also possible to organize educational events with Americans and people from Asia. It's much easier than we initially thought several years ago. And I don't want to waste too much of your time, but instead want to start with our first speaker, which is Amanda Cordell. And Amanda is chair and founder of the EOS network in the United Kingdom. And I've met Amanda on a Curate meeting in Cincinnati in 2019, and I was impressed by her energetic force for good, and as an advocate for patient education, patient rights, and any other way that may benefit the patients. And I also believe that Amanda is a parent of a patient with EoE, and I'm really excited that she will now present here on our symposium on patient perspective on EoE. And I'd like to ask Amanda if we can put the screen to Amanda. Yes, Amanda, we see you. And Ilse, can you put the screen sharing to Amanda as well? Yes, exactly.
Is that all good, as everybody can see that? Excellent. Thank you so much, Arjan. It's a real pleasure to be here this afternoon. As you say, my name is Amanda. I'm from EOS Network. We are a British-based charity with a global reach, and today I will be talking to you about EoE and the patient's perspective. So today, as Arjan says, I actually have two children who've been diagnosed with eosinophilic diseases, but I'm not going to talk about them today. Today I'm going to share you Stella's story and to demonstrate how EoE can have a lifelong impact. So Stella was born in the 1960s, a bygone era where you were told to eat what was put in front of you or go without. And this resulted in her often being told off for not finishing her food or eating slowly. And she was accused of being fussy and attention seeking. And this obviously isn't Stella in this picture, but she really felt that this demonstrated what she felt like as a child. As a teenager, she learnt coping strategies and she went on to train to be a chef to help control her diet. And this worked pretty well for her until she had children at 21. When her symptoms become much worse. So the symptoms are pretty much what we all see across the board with EoE. There's no surprises there. And the impact on her life was heavy, as you can see, which is no surprise either. She began the merry-go-round once she got to the point of feeling her symptoms needed to be addressed. And she went to the GP because she was avoiding so many foods, and was prescribed multiple antacids and PPIs, none of which helped. Eventually, her GP gave a referral to a gastroenterologist, and she went for a barium swallow and an endoscope, but they didn't find anything that actually changed her outcome. So she went back to the GP again for another go, and at this point he considered that maybe it was anxiety that was causing her problems and suggested that she had a mental health referral. Now, obviously, when she had a mental health referral, they assessed her and said, there's nothing wrong with you. This is obviously a physical problem, and we feel you need to go back to your GP. Well, this circle went on for 24 years, round and round, until eventually she attended the London Allergy Show in the hunt for extra foods on her free-from diets that she was trying. And this was 2011. And she heard about eosinophilic esophagitis, which sounded awfully familiar to what she was dealing with. And so she took this information armed off to her GP, of where they said they'd never heard of it and dismissed her concerns. Well, she carried on for another 5 or 6 years till eventually she moved home. And obviously in moving home, she gained a new GP and decided to approach the subject again because nothing had really changed in her life. She was still dealing with the same problems. Now, fortunately, this GP happened to have another doctor in the surgery whose wife had been dealing with similar symptoms, and they'd been investigating her for EoE. So they referred her, and surprise, surprise, the biopsies came back and she was diagnosed with EoE. Stella was now 50 at this point. The next couple of years was trial and error. She went back onto omeprazole again, a very high dose, which didn't agree with her and caused more gastro problems. She tried Montelukast, didn't seem to make any difference. She had a go at a 6-fed diet, which was even more than she'd already been cutting out. And she really struggled with that because there wasn't any extra support. And then they prescribed swallowed fluticasone. When I asked her about that, she said to me, "Oh, I don't know why it didn't work." And I said, "Did you were you told not to drink afterwards?" She said, "No, I was told to clean my teeth straight away." Well, she still was dealing with all her problems, and her diet was now to only 19 safe foods. So needless to say, her quality of life was pretty poor. And she went back to her specialist and they re-scoped her and referred her for an allergy referral. And after this, they said to her, we've pretty much done the extent of what we can do to help you. We think you need to go back to your GP again. And she appreciated them for their honesty, as it wasn't an area of their expertise. Well, things had moved on a bit in the last 4 years, and actually her GP said, "I think we can find you someone who really has expertise in this if you're prepared to travel." So she was referred to an EoE expert. Unfortunately, COVID got in the way for a period of time, but she eventually got to see this new specialist and they prescribed budesonide for her, which she didn't get on with either because she was sensitive to the honey with it. She's now currently trialling Julveza, and fingers crossed, we're pleased to say that so far she's seeing some promising results. Well, this whole experience for Stella has been lifelong, but we're pleased also to say that she's taken her experience and turned it into something positive. As she's no longer a chef, but she actually teaches other chefs and out places for food to actually cater for allergies and food intolerances. So after our experience, this is not an unusual experience within patients that we speak to, sadly. So we've started investigating some of these interesting learnings that we would like to know better about how the experiences have affected patients. The first thing we'd like to know more about is the length of time to diagnosis, as we see right across the board, multiple patients with 50 years to diagnosis, some with 10. So we took a look at our patient registration, and across the community we looked at 37 parents and carers, and our average across that period with children was 4 years of symptoms to diagnosis. And with an adult section, we took 57 adult patients, and sadly that is 9 years of symptoms to diagnosis. An area that we really want to focus on and make an impact of improvement. So during that 9 years of average, what are these patients actually experiencing? We recently put out a public survey in conjunction with Guts UK, asking adult patients what their experiences have been. So the first three is no surprise to many of you probably that are watching. All patients with EoE tend to experience some form of food sticking in the esophagus and pain, and obviously inevitably food avoidance and weight loss. But what we was most concerned about was the long-term effects, that nearly 3/4 had suffered a food bolus obstruction, 69% had suffered some form of stricturing and narrowing, and actually gone on, 31% of them had gone on to actually needing dilatation. So it's no surprise when one of the common quotes that we hear is that patients are incredibly scared to eat in front of others in case they choke. So how does this affect outside of these symptoms? How does the real impact on their quality of life? Well, EoE affects every aspect of a patient's life, as we can see from this graph here. Social life, incredibly difficult. Over half of our patients we spoke to had made a major impact. Eating, travel, going on holidays, diet, the type of diet, well, we know that that's a big problem. Work, and most concerningly, coping with the symptoms of it and quality of life for nearly 3/4 of our survey, and a financial impact too. So how are the treatments that are being used currently working for patients and carers? 34 out of the 39 that were surveyed in our public survey had actually been prescribed proton pump inhibitors. Now, only 19% were satisfied with this as a treatment for their EoE, and 74% didn't feel this improved their quality of life, although some did mention that although it didn't improve their EoE, it had some effect on reflux symptoms. 18% had reported adverse side effects, which is quite a concern too. Food elimination, well, we all know this is a real difficult one for patients to manage. 28 out of the 39 had attempted some form of food elimination diet, and you can see the varied types that they'd accessed from the graph on the right. 23% were satisfied with this as a treatment. More than 25% of them had tried to eliminate more than 6 foods, which is really concerning. And 67% found it difficult to be compliant when away from home, which is no surprise. But the big concern to us, and one that we wasn't shocked by really, is that 33% of our surveyors found that they were struggling to get dietetic support. And obviously we know the impact that being on a restricted diet has, and that is that food is the most social thing that we do as a human being. So when you can't eat what other people are eating, it has a massive impact on your life and can be incredibly isolating. We also found that 28 out of 39 had been prescribed some form of swallowed fluticasone or a budesonide slurry. 30% of patients were actually happy with this as a treatment for their EoE, but 59% didn't feel it improved their quality of life. Now, we also know that over a quarter here found it really difficult to follow the instructions, which reflects many of the comments of our patients. They go home with a leaflet, it doesn't say what they need to do, and it's very much more luck than judgment how they get that treatment to work. And 19% found it incredibly difficult to manage when away from home, actually administering these types of medications. And you can see obviously the concern that most patients had was actually whether they were getting the right amount of medication or whether they were doing it properly. So obviously we're looking to Jurvesa become available on the market in Europe and other places around the globe. Now, we had a very small cohort of patients that actually responded to this survey because they are struggling to actually get that in the UK at this moment in time. Only 10 of our 39 had actually been prescribed Jurvesa. 60% of those, so 6 patients, said they were very satisfied with it as a treatment for their EoE. 20% of them felt it didn't improve their quality of life. And 30% of those 10 had struggled to get it from their doctor or pharmacist. So you can see it's a large percentage of people are struggling to actually get that medication still now, which is part of what we use this survey for as submissions for HTA. And I'm delighted to say that today the Scottish board have approved Jurvesa as an acute treatment and maintenance, which is breaking news. So at EOS Network, what are we doing to actually change this outcome for patients, and what opportunities are there for you as healthcare professionals? So you can see from this slide, which is incredibly busy, that's what we're up to, which is an awful lot at the same time, but we really are a platform to connect all of these relevant dots, and that's exactly what we wanted to happen. We have 2 arms to the organisation. We have the community hub, which is there to support patients, and we work locally with patients. We also direct for more local support if it's available. We also work with global rare disease organisations such as obviously EUREOS today, CGEAR, EURODIS, and we also work with pharma and industry to try and help with bringing new medications to market and for disease awareness and obviously for medical education. And we also work with other medical associations such as the British Society of Gastroenterology, BISCAN, etc. And we're working with those to build new guidelines to increase the medical education, which we can then disseminate amongst our professionals' global network, which is there really to help you communicate between each other, to help disseminate the information such as what EUREOS is bringing together, and to bring collaboration and hopefully improve access for patients to medical care. So we would suggest today that we would like to invite you all to register with our Professionals Global Network. By joining, you'll have an opportunity to receive important news updates and research updates from around the globe. You'll also have access to our LinkedIn group forum, which will give you a space to connect and collaborate and talk about all the great work that EUREOS is doing and CGEAR is doing, etc., around the globe. And it will also give you a place to recruit the clinical trials or your surveys and to list your publications. And of course, importantly, access to patient resources. I'd like to thank you all for taking the time to listen today. And if you have any questions, I'd be very glad to answer them for you.
Thank you, Amanda. That was a fantastic presentation. It really provides also a new scope on how sometimes how our patients see us and how we can how we see the patients and the disease. And there are a few questions from people in the audience that I would like to ask you. And here's one question from Paula Hint, and she is a dietitian at St. George's. And she says, "Dear Amanda, could you please share your data with me if possible? I really like to help us with the EoE patients at St. George's. And I'd like to suggest that we share your presentation together with the other presentations on the EUREOS website. And we can also send you a copy for the EOS network website."
I think that's an excellent idea.
Yeah, so maximally disseminated. And here's another comment from Stefan Mielke. He says, "A fantastic presentation by Amanda. Is it possible to have access to the results of the patient survey?"
We are looking to put it together as a publication, actually. I was very grateful. We worked in conjunction with Guts UK to get as much spread as possible. And it was very nice to give us an opportunity to make it public, so we weren't getting any biased opinion from anywhere. And it was a great insight. We felt it was a great insight to patients. You know, obviously we speak to people on an individual basis, but when you get that across a broad spectrum, and it won't be the last survey that we do. So if anybody's got any suggestions that they would like us to put out to our patients, we're very willing to listen to that.
Yeah, there are a lot of questions and comments now coming in. Here's from Margaret Collins saying, "Thank you, Amanda. Did the patient you highlighted in the beginning have a stricture diagnosis?"
Right at the beginning. So are we talking about the?
I think the lady with the delay.
The lady with the delay. I don't know. I can find out for you. I think actually she'd made herself sick an awful lot in her lifetime and learned that as a coping mechanism. And she's also been on a highly restricted diet all of her life.
Okay. And here's a comment from Alex Strauman, our society's founder. He says, "Dear Amanda, congratulations on this excellent presentation, Alex." And it's another thank you from Paula Hint. And here's also, let me see, a comment from Luc Biederman. He says, "Thank you so much, Amanda, for this beautiful and impressive talk. Officially, he says EoE was the first described end of the 80s, and it hardly existed prior to that, at least wasn't described, at least in the medical books." And then he says, "Have you met more patients where the disease may have started considerably earlier?" Do we have to assume that
I have.
EoE has been existing much earlier, maybe several decades before the first descriptions? And he asks
Off
you,
the top of my head, sorry.
Yeah, he says, what's the
I can,
earliest presumptive initial EoE manifestations that you recall from patients that have shared with you?
I have at least, off the top of my head, I have at least 4 patients that have had 50 years of symptoms.
Oh, that's really impressive. That also changes a bit of our history on EoE. I think Luc will get in contact with you about it. Maybe we should consider describing these patients and see if they agree. Contact them and see if we can learn from their stories. Well, there's another 10 more questions, but we're running out of time, and we have to go to the next speaker. I'm really impressed by the presentation, and thank you so much for making the time to share this with us.
I'm very willing to share my email address, or alternatively, if you just go to the website and click on the contact us or join us, then please feel free to submit your questions or get in touch. I'll be delighted to hear from you.
Thank you so much. Thank you, Amanda. And we're gonna continue with our next presentation, which will be done by Salvatore Olivia. And Salvatore is a pediatric gastroenterologist working as assistant professor in pediatric gastroenterology at La Sapienza, the University of Rome. And he is a new EUREOS steering committee member, approved of 30 minutes ago. And I personally hope EUREOS can become an EoE organization where adult gastroenterologists, pediatric gastroenterologists, allergists, basic scientists, dietitians, we can all meet and work together on eosinophilic gastrointestinal diseases. And Salvatore is coordinating the European Perspective Pediatric Registry of EoE. It's called, I think, PEER. And he will present on the learnings from the Pediatric EoE Registry. Salvatore, please.
Good evening, everybody. Thank you for this kind presentation, Arya. I'm glad to be here, taking part in this important EUREOS webinar. And as I mentioned by Aryan, I would like to share our experience as a pediatrician in managing data about the EoE in pediatric age. And I'm also glad to have my talk just after the Amanda's talk, which is very interesting in terms of symptoms and patient experience. And before going in depth of the talk of today, I would like to remind you that in children, we have a wide variability in terms of clinical presentation, as also presented in the Amanda's talk. So in particular, in younger kids, it's very difficult to identify those kids needing for endoscopic evaluation, because we have a considerable overlapping in terms of symptoms with other conditions much more common than EoE, for example, GERD or food allergy or just functional abdominal pain. While in older kids, we usually have symptoms that are more similar to the adult symptoms and are more suggesting EoE, for example, dysphagia or food impaction. So unfortunately, there are a lack of data in terms of clinical presentation of EoE, in particular in children. Recently, the North American network has published their data about the COFAR registry, a multi-center registry, including and describing for the first time the EoE North American population. And actually, the ASPGAN, our pediatric society, did the same in a retrospective way by the retrospective registry led by Noam Zevid, publishing a couple of papers and describing for the first time the pediatric European population. But there are no data in terms of prospective description of EoE in children, except for those data presented by the two last paper of the EoE Connect registry. So for this reason, the Eosinophilic Gastrointestinal Diseases Working Group of ESPGHAN has set this registry with the aim to collect prospective data from EoE children in Europe. And the other aim was also to assess the compliance with the old diagnostic criteria, which means the presence of a double endoscopy and the PPI trial in a real practice setting. And finally, also to increase the cooperation among the different European referral centers for children. So in this registry that started in 2011, we included any patient with a peak eosinophil count more than 15 eosinophils per high power field, and that was considered for the complete diagnostic workup, including the PPI test and the second endoscopy.
Salvatore, can you put your screen on the presentation mode?
Okay, but you cannot see the slides.
Yeah, if you put it in presentation mode, it is easier for us to see it.
Okay, let me try again.
Below on the presentation mode in PowerPoint.
Yes, but it was in that way. I don't know what
and
is
it was
happening.
somehow frozen.
Okay, let me try again, because I think I was sharing only the PowerPoint. So if I share the main screen, it should be better.
That one.
That way.
Yes.
Okay.
This is much better.
Okay.
Thank you.
Okay. Okay. I didn't know. Thank you. So as mentioned, this registry is a multi-center prospective registry based on an online platform with a dedicated website in which investigators can include their patient. There was a case anonymization before including any patient, and each case can be identified only by the local investigator. There were a symptoms report as well as the report for endoscopic findings and the peak eosinophil count over the entire esophagus. At the end of the diagnostic process, it was possible to select the type of treatment that was used for that specific patient, and finally, the diagnosis suggested by the local investigator. So there was even a report for the allergy evaluation, as well as a box to include the pH study test performed at the beginning of the diagnostic process. During December 2018, we analyzed the cohort of patients available at that time in our registry for 582 patients analyzed. And here you have the number of participating sites and the number of patients for each specific country. Looking at the results, as you can see, the majority of patients were male with a median age of 10.5 years. As mentioned by Amanda during her talk, as you can see, there is even in this cohort a considerable discrepancy between the onset of symptoms and the time of diagnosis, with the diagnostic delay of 1.2 years as a median value. And this diagnostic delay decrease in presence of a concomitant food allergy, while considerably increase for younger kids less than 6 years of age. You see here also the atopic comorbidities and the family history of allergy for our cohort of patients. And looking in details, you see that the most common allergy associated with the EoE was the food allergy. Moving on, symptoms, the majority of patients had GERD-like symptoms, followed by dysphagia, food impaction, vomiting, and so on. It is interesting to note that in 5% of cases, the EoE diagnosis was an incidental finding during an endoscopy performed for other reasons, such as celiac disease or something like that. We did not observe any seasonality in terms of clinical presentation and number of diagnoses over time, while we wanted also to look at the symptom distribution according to the age. So we divided our cohort according to the median age in two groups, younger kids and older kids. And as you can see, growth failure and vomiting were more common in younger kids, while dysphagia and food impaction were much more associated with older ages. And this difference was statistically significant. Looking at the endoscopy, the majority of patients had inflammatory findings such as exudate and furrows, while rings and fibrotic lesions were less common. And in particular, we did not observe in this cohort cases of stricture, which seems a very rare event in children. It is also important to note that about 17% of patients presented a normal endoscopy after the first and the second evaluation. And this is particularly important because without a specific histological examination, it was not possible to identify those patients. Looking at the endoscopic finding distribution by age, as you can see, the inflammatory lesions were much more common in younger kids, while fibrotic lesions were more described in older kids. While the normal mucosa was equally distributed among the two groups. We did not observe any difference in terms of peak eosinophil count over the entire esophagus, while about 20% of patients perform a pH study with the positive results only in 40 of them. As mentioned, one of the aims of the registry was to assess the rate of PPI responders. As you can see in this course, 26% were responders to the PPI therapy, while the remaining were considered for topical steroids, diet, or a combination of one or more treatments. So we wanted to compare the PPI responders to the real EoE according to the old definition of the disease. As you can see, we found that the majority of PPI responders were older and male compared to the real EoE patients, and they also had less severe lesion compared to the EoE patient. But unfortunately, none of these individual factors was independently predictive of the PPI response. So as you know, the definition of the disease has changed after the agreed consensus in 2019. So we wanted to change the aim of the registry from the diagnostic phase to the natural history and to the maintenance phase. So we tried to consider from 2019 all patients with the EoE diagnosis, regardless of the presence of a PPI test, as stated by the new guidelines. And we also asked the investigator to include any endoscopic follow-up over time in order to have also information about the maintenance phase. And we wanted to objectively assess the disease by using a validated score for symptoms, histology, and endoscopy as well. Looking at the data analyzed during December 2019, as you can see, we had 512 patients with more than one follow-up after the induction phase. the majority of them were also male, and for a total of 3,018 endoscopy, and with a median number of endoscopy per patient of 3.5, and with the median follow-up time of 2.6 years. Moving on, the maintenance strategy utilized by the investigator, as you can see, the majority of patients were receiving PPI followed by diet and topical steroids, regardless the type of the induction therapy. The majority of them were receiving a monotherapy, while about 40% of them received a combination therapy of two strategies, and a very small proportion of patients received all the three strategies at the same time. Looking at the efficacy as a monotherapy in terms of combined remission, which means both symptoms and histology, we found that PPI were less effective than topical steroids and diet at the first follow-up after the induction phase, but all the three strategies were more efficient in maintaining remission compared to the absence of therapy that was able to maintain remission only in 16% of a patient without treatment. Looking at the comparison between monotherapy versus combined therapy, as you can see, in general, on the total number of follow-ups we considered, combination therapy obtained better results than the monotherapy in terms of symptoms, endoscopy, and histology as well. So in conclusion, I would like to run through this following point. As mentioned at the beginning of my presentation, in children, there is a wide variability in terms of clinical manifestation of the disease. And this leads often to a diagnostic delay that can be influenced by the age of patient or the presence of atopic comorbidities. And this is important to remember that a normal endoscopy, especially in children, does not exclude the presence of EoE. Unfortunately, that the presence of stricture is a rare event, at least in the pediatric age. And unfortunately, in our case, it was not possible to identify any predictive factor able to detect the PPI responsiveness over time. And according to this data, a wide variability also exists in terms of maintenance strategy, and this should be clarified in the next future. But according to the data, PPI seems less effective, while combination therapy seems to obtain a better result than the monotherapy. And finally, I would like to thank all the investigators participating in this registry, and in particular to Georgia Mildias and Noam Zevit, who initially developed this registry. And this is the last update during June 2020, with about 728 patients enrolled from 24 sites. I'd like to thank you all for listening to this presentation. If you want to receive more information about the pediatric registry, feel free to text me or visit the website. Thank you.
Yes, thank you so much, Salvatore. That was an excellent presentation, and I really enjoyed at these data, and we could compare it with the adult registry. Now, there's several questions from the audience here. This Paula Hint asks us. She says, "I'm really interested in the incidence of malnutrition and deficiencies in children with EoE. Is there any data on that?"
Unfortunately, we do not have clear data about that. We try to analyze if there was some malnutrition, but in our registry it was not initially considered as an aim, so it is not possible to have clear data about our population.
And here's another question from Michael Veet, and he's a pathologist, and he This question is, can you relate the success of therapy, especially modal therapy, with the distribution pattern of the eosinophils?
Yes, this is an interesting question. I try to analyze if those patients receiving combination therapy had better results in terms of decrease of peak eosinophil count over time, or had a more severe disease at the beginning, but it was not possible to find any difference. One of the main issues in the analysis of the registry is that we had a really big variability in terms of follow-up strategies and even findings of sometimes also missed data. So it's very difficult to standardize the evaluation. So I would like to answer that question, but it's still not possible so far.
Okay, and here's one other question. If PPI response is so poor, how will this impact the recommendation for future treatment algorithms in children?
So far, PPI, diet, and the steroids are considered on the same level as a first choice therapy. So I would consider in the same way even now. My feeling, and even the data shows that PPI could be less effective, but we don't know at the beginning of the story if PPI will work or not. So I think we should focus on some predictive factor able to detect the PPI responsiveness.
Yeah, to select which patient you will start what treatment. Yeah, okay. With that, I would like to thank you, Salvatore. That was an excellent presentation, and we're gonna move on with the last speaker of today, which is Marc Rothenberg. Marc, please start your camera, and I will announce you. Marc is Director of the Division of Allergy and Immunology and Professor of Pediatrics at Cincinnati Children's Hospital. And Marc is really a giant in EGIDS with numerous original papers, mainly basic science papers about the pathophysiology of EoE and eosinophilic gastritis and gastroenteritis. And I don't think there is anyone listening today that doesn't know Marc. And Marc, we're really excited to have you here today and present on this quite a hot new topic, eosinophilic gastroenteritis, new insights into diagnosis and treatment. And it would be great if you could start your camera as well. We see your screen, but not your camera yet.
Yes, I gotta hold up on the camera because when I put it on, it said the internet connection was weak. So maybe better that way. Do I sound all right?
Yeah, we can hear you.
Yeah. Okay, terrific. And you can see the screen?
Yeah, we can see the screen.
Okay. Arjun, thank you very much for inviting me to give this talk to EuroEOS. It's a pleasure to come and show you our work focused on eosinophilic gastritis and gastroenteritis with a focus on new insights into diagnosis and treatment. The work today really focuses on the next slide, which I'd like to be able to show. Anyhow, the work focuses on work that we've been conducting through the Consortium of Eosinophilic GI Disease Researchers, which is a national consortium in the US. It's supported by the US, composed of about 15 sites, whose focus is to improve the lives of individuals with eosinophilic gastrointestinal diseases through innovative research, clinical expertise, and education via collaborations between scientists, healthcare providers, patients, and professional organizations. SEGAR involves a number of different working parts. We have something called the Data Monitoring and Coordinating Center that monitors our data. This is a centralized research resource from the NIH that allows us to analyze and maintain the data. We have domestic sites in the US that participate in the research. We have consultants as well, primarily in Switzerland. We have an external advisory board with some distinguished doctors in gastroenterology and allergy and pathology and other fields that give us direction. We very strongly partner with PAGs, which are patient advocacy groups, which are critically involved in the research. And we also work together with patients through the PAGs and also independent of the PAGs, particularly with the clinical studies. And we also partner with professional societies, and that's where our relationship with EUREOS is part of the SIGA operation. And we have dialogue and common interests, and I think this seminar today, this short lecture, may be able to be an impetus for further activities between SIGA and EUREOS. And of course, we work with the NIH, and it's funded through an organization, an old grant called the U54, which is basically a cooperative mechanism, so that the grant isn't just us working together, but we're cooperating with NIH experts to advance this set of rare diseases. The research focuses on a number of different components that are dynamic and moving. We have an observational study under clinical care, and we're following our patients with all forms of EGID. We are focused on disease classification. We're not only looking at the cellular and molecular components, but also the disease definitions. We're working to improve diagnosis and prognosis, and the talk today will primarily focus on that. We also are focusing on positioning EGID as a directive from the NIH so that it's readily available for clinical trials, an area that they focus in on for rare disease research, focused on clinical trial readiness, developing things like the PROs and molecular panels, which I'll talk about, that allow us to quickly move into clinical trials and hopefully advance the field so these are available for others. And we're also looking at targeted treatments aiming to improve outcomes. So the talk today is focused on eosinophilic gastritis, and it's nice to move down the GI tract from the esophagus. Most of the work does focus on EoE because of its prevalence. You can see here estimated prevalence out of 100,000 individuals. EoE would be about 50 individuals, where EG is about 6.3, and EC is even less than that. The diagnosis of EG, like EoE, is dependent upon endoscopic procurement of biopsies, analysis histologically by the pathologists, and enumeration of eosinophils. There is not a fully agreed upon threshold for diagnosing the disease, but SIGA has agreed to use the 30 eosinophils per high power field and at least 5 high power fields for diagnostic purposes for research investigation into this disease. This is a moving target, and some of the work I'll show you today is helping us provide tools and understandings of the changes that take place at different levels of eosinophils in the stomach. And these numbers are based on the calculated averages as well as the maximum that we've seen, and this is the range that's been reported in some studies, particularly in the stomach. Substantial advancement in EoE has taken place through endoscopic biopsies and analysis of molecular transcript profiles. And we, along with other groups, have defined the EoE diagnostic panel, which is a set of transcriptomes that are abnormally expressed in the esophagus, and allows us to probe very rapidly the molecular and cellular components of the disease. And this has truly advanced the understanding of the disease so that we now have targeted therapy available, which I'll come back to at the end. But what I'd like to focus on today is how we've also begun to use this type of approach in the stomach. We reported a number of years ago the EG transcriptome. And this is a set of hundreds of transcripts that are differentially expressed in the stomach of EG patients compared to EoE. One of the early remarkable findings that we had was that there was only 7% overlap between this transcriptome, EoE, and the gastric transcriptome EG patients. There was overlap of some of the cardinal pathways, particularly the type 2 immune reaction with the IL-13 and IL-4 and eosinophil signature genes, as well as mast cells. But we wanted to advance the diagnosis of EG by further probing the EG transcriptome. And this is work that we did through SIGA, where we looked at a number of gastric samples across the SIGA sites, and we identified through RNA sequencing a series of genes that were consistently abnormally expressed in EG compared to controls, and this was independent of age of patient, atopic status, site of where this in the stomach versus the different locations, the antrum of the body, and also independent of which institutional gastroenterologist was biopsying. And we came up with a panel of 48 genes that we were able to narrow down that really allowed us to differentiate EG from the control populations. And we even went further to identify 18 genes, which now referred to as an EGD18 panel, an EG diagnostic panel with 18 genes. And what you can see here are the genes that are either upregulated or downregulated in the disease compared to the control. And then you can see in this diagram over on the right, the PCA plots of the patient's EGDP score, and you can see a complete separation of patients with active EG compared to non-EG, and other controls were showing similar patterns, and we were able to replicate this in independent cohorts as well. So this allowed us to develop a scoring system based on molecular profiles, which we refer to as the EGDP18, which is really a summation of the delta CT values of the most highly dysregulated 18 genes. This scoring system does directly correlate with the peak gastric eosinophil levels. You can see a very strong R. It's an inverse association, because the lower the EGDP score, the higher degree of abnormal changes. So you can see a very striking correlation. We can also see that we can follow patients longitudinally, probing them with the molecular profile, and this allows us to differentiate patients with active versus inactive disease. And as I mentioned earlier, there's a very strong correlation and consistency between the gastric body and antrum. So we've used the EGDP score to probe the histological and endoscopic features of the disease. And in this slide, we are looking at various EG histological features that are now being used as part of an EG-HSS histological scoring system that our pathology experts have developed, particularly Dr. Collins and her team. And this shows you the various scores of these different parameters besides the eosinophil levels and their relationship to the delta CT value of the EGDP score. And you can see that there were some features of the disease, such as the eosinophils in the muscularis and the lamina propria smooth muscle hyperplasia, that didn't have a very substantial correlation. But then there were others, like the periglandular circumferential collars, that showed a very strong association with the molecular changes, and of course others as well, as indicated on the right what they were. We've also been able to analyze the molecular profile as a function of the endoscopic changes. And these are different parameters that we've been tracking in EG: granularity, nodularity, erythema, friability, folds, and erosion and ulcers. And you can see that, again, similarly, the log 10 FDR corrected p-value does show variations depending upon which feature we're looking at. But the most striking ones was granularity, which is shown in the image on the right, and nodularity, which is the micronodules that are characteristically seen in this disease. And they showed very striking and strong association with molecular patterning. And of course, we know particularly what genes are associated with each of these, and that's been recently reported by an article by Schroeder et al. And I refer you to that for the details of what genes they are. Turning back to the diagnosis, we have also been interested, as a major amount of our focus is on developing less invasive and non-invasive platforms. And in EoE, the search continues. There's certainly less invasive testing that's being developed by a number of means, but there's very little success so far with blood markers in EoE that are clinically relevant. Now in EG, we have some encouraging data. This is looking at both plasma and serum from a series of patients and looking at a series of relevant proteins that we identified from a multiplex assay. And this is focusing on ones that showed some promise of particularly biological functional associations with the disease. And what you can see here is the minus log 10 of the adjusted p-value. So these are some very substantial associations between the eosinophil levels and the diagnosis of the disease, and these particular cytokines that we can measure in the plasma and the serum. And you can see that eotaxin 3 is very strongly associated in the plasma, which is stronger than the serum, but both areas. TSLP is very positive in the serum, and IL-5 is a very nice correlation as well in both the plasma and the serum. Eotaxin 3, similar to the esophagus, is one of the chief genes that's overexpressed in the stomach of patients with EG, probably related to the intense eosinophil ilia that we see. And you can see in the lower data sets that these have individually pretty nice sensitivities and specificities. And this combined allowed us to develop an EG score, which is a 4-point score. It's based on the plasma or the serum. We prefer the plasma because it's got a high yield. And this allows us to score the plasma samples from these patients and determine their degree of correlation and diagnostic potential. And what you can see here is that the blood EG score does in fact predict disease activity. In the plasma on the upper left, you can see the EG score in the plasma differentiates patients from active EG from non-EG. There is some overlap, so it's not a perfect score. And you can see that the serum markers as well differentiate them, as well as the plasma EG score differentiates active from inactive, and then also the serum EG score shows similar trends. We've been able to construct receiver operator curves for these plasma and serum biomarkers. And what you can see here is the individual relationships between individual proteins in the plasma or the serum, and their very good sensitivity and specificity based on the area of the curve. But I want to draw your attention to the combination score between these three markers. And what you can see here is in the red line, an area under the curve that's approaching over 0.9, in this case, very nice plasma EG score using this combined score, which we think shows great potential to monitor and diagnose the disease, but particularly for long-term monitoring of the disease. And this is consistent with data that we had in the past that the blood eosinophil level was also correlative in EG, more so than we typically see in EoE. So in summary, what I've shown you so far today is that we've been able to use molecular endoscopic, histological, and circulating biomarker-based discovery to develop diagnostic panels for EG involving multi-site studies. The sites involve 185 patients with multiple sites across the US, and we've been able to develop tissue-based profiling based on the 18 significant genes which monitor the disease activity, and they improve the diagnostic chance, particularly for ambiguous cases that wasn't shown today, and also do correlate with histology and endoscopy. As scientifically, which hasn't really been carefully gone over today, these do, however, also inform information about the pathogenesis of the disease, which is going to help us with development of therapy. And the blood-based markers are very promising, particularly the eotaxin 3, IL-5 TARC, and TSLP. So I just want to close by talking to you about potential therapy for this disease. And this data is old data that we developed a few decades ago, actually, early work that I had done with Simon Hogan, and he was a postdoc in my lab, where we developed a model for eosinophilic GI disease in mice. And we showed that these mice developed a number of features of eosinophil GI disease, particularly in the stomach. And this is looking at the size of the stomach in these mice that were challenged with the allergen. And you can see these mice developed gastromegaly. And this was shown to be due to eosinophil infiltration and a degree of dysmotility that was occurring in the stomach. And at that time, we didn't have eosinophil knockout mice, but we had was eotaxin knockout mice. And these mice were greatly devoid of eosinophils in the stomach, and you can see that they were protected against the gastromegaly, which we linked to external necrosis taking place that was potentially mediated by the eosinophils. So this was some of the early data that established the potential causal role for eosinophils in this disease pathology. We also have seen, as I mentioned earlier, that there's a correlation between the peak eosinophils in the blood and the tissue. More recently, we have conducted a study looking at eosinophil knockout in humans, and this is where a drug called benralizumab. So these patients take this drug, which mediates antibody-dependent cellular cytotoxicity. We've conducted this study, which is a single-site, double-blind study with 24 patients, and half got placebo and half got drug. The study is closed. We're analyzing the data. The data is still being cleaned, but we have an open label extension, and we can tell you that, very interestingly, that the eosinophils are depleted in nearly all the patients. And some patients, like subject 1, have a complete resolution of the eosinophils and a normalization of the histology, a very dramatic change and a clinical benefit as well. Whereas other subjects, we can see here, have a huge amount of eosinophils. They are depleted by the antibody, but the patients still have active inflammation that's non-eosinophilic, that's ongoing. Now, these are patients that had atopic EG that appeared to be very similar to this patient over here. So what we're seeing is already in this early data is there's going to be heterogeneity in eosinophilic gastroenteritis in humans, and I predict that some will be independent of eosinophils in terms of the pathophysiology and even the clinical symptomatology. So I'd like to conclude what I've shown you today is that the EGDP score, which only includes 18 genes, was sufficient to diagnose EG cases. The EG molecular profile also strongly correlates particular endoscopic and histological features, thus providing insight into the pathogenesis of EG. Circulating biomarkers reflect local changes in the stomach, potentially serving as an early non-invasive test. The biomarker profile substantiates the involvement of type 2 immunity, and the therapeutic precision strategies that are currently being pursued include eosinophil depletion with the antibody benralizumab, another antibody that shows great promise based on company open release data called larentelimab, which is an antibody that also depletes eosinophils by a different mechanism, shows very strong promise in EG in a phase 2 study. And we also have anti-type 2 cytokine therapy, which is mainly through IL-13 and IL-4 drugs like dupilumab, and also specific antibodies against IL-13, which show great promise as well. So I'd like to close by thanking all the people who contributed, particularly reminding you about CIGA, the Consortium of Eosinophilic GI Disease Researchers. This is supported by 3 different institutes at the NIH. It's part of the Rare Disease Clinical Research Network, and we also have a very strong partnership with various patient advocacy groups. Thank you for your attention. I'll be happy to answer some questions.
Thank you so much, Marc. And perhaps we can now put your camera on for the discussion.
Yeah, so let's try that.
We'd all like to see you. Thanks, that's great. I'm waiting for the questions to get in. I personally have, I was surprised that only 7% of the transcriptome between EoE and EG were actually overlapping. Did you expect this, or was it a surprise to you as well?
It was a surprise to us, but when we think about it, the gastric transcriptome itself versus the esophageal transcriptome, there's a dramatic difference in the genes that are normally expressed in the squamous epithelium, the esophagus, versus a columnar and more complex epithelium involving multiple different cell types in the stomach. We actually went back and we looked based on that at eosinophilic allergies outside of the GI tract, atopic dermatitis versus asthma, and we compared atopic dermatitis as well to EoE. And we found more overlap than actually EG, but the overlap was actually mainly about 10 to 20% overlap. So even in classic atopic dermatitis, there's going to be a very modest overlap with asthma, and EoE does more align with atopic derm, but not as much as either. And I think what this is telling us, and it comes back to some of the work that we have done with the genetics of EoE, some of the causes for EoE are related to esophageal specific genes like helpine 14. And we don't yet know the cause for eosinophilic GI disease outside of the esophagus, at least from a genetic point of view. But I predict that we will find that there are other tissue-specific elements that are interacting with the atopic genes, like the TSLP locus, which is another driving force, more of an atopic predisposition.
Yes, thanks. Here's another question, and that is let me see. Oh yeah, the patients in the benralizumab group that responded basically with all inflammation and symptoms compared to the ones that did not respond or just had the eosinophil depletion but no symptom or inflammation reduction. Did these patients differ in any other way transcriptome or other characteristics?
We are currently looking at that. We have not found any distinguishing features, but it's a high priority area.
Okay.
It's a high priority subject. We haven't found anything yet to distinguish them.
Yeah. Here's a question from Iku Irano. He asks you, does the plasma biomarker activity correlate with presence of peripheral eosinophilia?
Yes, it does correlate. There is, we looked at that as well, and there are correlations, particularly with some of the cytokines, but also with the molecular profiling in the stomach does correlate with some of the peripheral changes as well.
Okay. And here's a question from Alex Trauman. He asks you if EGDP score correlates with
heavy hitters online today. Hi, Alex and Eco.
He says, if the EGDP score correlates with eosinophils, what does it add in addition?
Well, when you look at eosinophils, as we know, you're looking at one cell type, and you're looking typically at an enumeration of that cell. And it turns out, in the case, for example, in EoE, where we know more about it, that we've been able to define at least three different endotypes, and those endotypes have different clinical features, and we believe different prognostic indications. And the most classic one, which would be interesting, obviously, to you, Alex, is the fibrostenotic phenotype that you've worked so elegantly on. And it turns out in our hands, and be interested in yours as well, that the eosinophil levels are not different between all of these different endotypes. And we find, and we're seeing very similar findings with the type 2 cytokines, the other paper we just published by Dunn et al., that there's a lot of heterogeneity in EoE, and we believe also in EG, although EG is not as advanced, but we're predicting that the eosinophil levels are going to be fairly unrelated to some of the other features. And the molecular profiling is giving us a lot more heterogeneity and understanding of the disease and the variability in the patients, and we believe will ultimately prove to be valuable for different biological treatments that interfere with this targeted pathways that are involved selectively in different patients.
Okay, and here's the final question. In the trial with AK002, there were several patients that were included. They had dyspeptic symptoms and eosinophilia on histology. And I personally must admit that often if a patient has dyspeptic symptoms, we do not take biopsies from the stomach. And some of the patients did not have really pronounced abnormalities on endoscopy. So I assume that there's many patients that have dyspeptic symptoms, which is related to EG actually. And there's a chance they'll respond to therapy, anti-eosinophil treatment. Well, we never expected this. What's your view on that?
Yeah. Who's this question from?
Yeah, it's my own question.
Oh, your own question. I think that that's a very interesting point. And with the this study that you mentioned, the Alico's study, that was indeed seen that there were patients that were actually de novo patients that were diagnosed initially with, like you said, dyspeptic symptoms, and they responded to the drug. They did have elevated eosinophils, and that was surprising to me and probably to many people. There would be such a high rate of responsiveness in these patients that weren't previously diagnosed with eosinophilic disease.
I didn't expect that. So I thought these were dyspepsia patients with some non-relevant eosinophilia, and I didn't expect them to respond to the drug. Now, because they respond, yes, I assume that these eosinophils play a role in their symptom generation.
That would be an assumption to make. The cyclic antibody also affects mast cells, so it's complicated, but it's very interesting and it's important work, and it's very exciting opportunity for potentially helping patients, but more science is needed.
Yeah, so I assume that if we start to biopsy more, now there's in the future treatments for these patients, we have to biopsy more probably.
Right.
Yeah.
Right.
Yeah. Okay, Marc, we're gonna
do a lot of biopsies. To get these diseases diagnosed, you obviously, like you said, have to biopsy. But some of these studies, you know, are very, you might call aggressive with biopsies. You know, like we're designing one now and it involves 20 biopsies. And that's what my colleagues, the GI doctors that I work with, routinely do when they're working up these types of patients. So some might consider that to be normal, but others might consider that to be a lot. But it's important to know that we need a lot of biopsies to get to these diagnoses established.
Okay, Marc, I'd like to thank you again for your contribution to this scientific symposium. Thank you so much.
Welcome.
And also,
good
like
luck
to
with this. I hope that we'll bridge together things related to SIGA and EUREOS. Thank you. Have a nice day, everyone.
Thank you. And also thank you to Amanda Cordell and Salvatore Olivia and to Laurie, who made this logistically possible. And with this, I'd like to conclude our webinar of today. Thank you.